Our thread probes and protocols enable a rapid and sensitive quantification of free and surface-bound elastase and cathepsin G activity in sputum from patients with neutrophilic airway diseases.
These methods make it possible to explore different aspects of proteases pathophysiology. Plate reader measurements permit for large screenings. Confocal microscopy visualizes enzyme activity with subcellular resolution, while flow cytometry enables single-cell phenotyping and protease activity quantification in a personalized fashion.
This technique can be expanded to different airway diseases, such as cystic fibrosis, bronchiectasis, or COPD. And it can be adapted to different biosamples, such as blood or bronchoalveolar lavage or mice samples.
Before starting the sputum induction procedure, inhale 200 micrograms of the beta-2 receptor antagonist salbutamol. Afterwards, inhale hypertonic 6% saline solution for 15 minutes using a nebulizer. Collect the expectorated sputum in a petri dish. Separate mucus clumps from the saliva into a petri dish with the help of a pipette tip. Weigh the mucus, then add 4 volume parts per weight of 10% sputolysin in PBS to the sputum. Incubate the mixture at room temperature on a rocking shaker for 15 minutes to dissolve the mucus. Quench the reaction by adding 1 milliliter of cold PBS for each milliliter of 10% sputolysin.